Right now, inside your body, a microscopic, almost completely silent biological paradox might actually be taking place.
Yeah, it's a terrifying thought when you really break it down.
It really is. Like your skeleton could literally be slowly dissolving,
Hmm.
just, starving for calcium, growing brittle and weak over time.
Right.
And while that is happening, while your bones are losing that structural integrity, the calcium that should be fortifying those bones is instead depositing itself into the soft, flexible tissues of your cardiovascular system.
Exactly. It's moving into the completely wrong places.
Right, like it's literally turning your heart's arteries into rigid, solid bone. And, this isn't science fiction.
No, not at all.
It is the biological reality for millions of people experiencing the simultaneous progression of osteoporosis and cardiovascular disease.
And for decades, I mean, the medical establishment treated these as completely separate issues.
Right, just two completely inevitable consequences of aging that had nothing to do with each other.
Like you just get weak bones and stiff arteries, and that's just getting old.
Exactly. But today, we are going to explore the single molecular traffic cop that prevents this fatal misdirection of calcium from happening in the first place.
It is such a crucial piece of the puzzle.
It really is. We are jumping into a deep dive on a severely underrated, just highly misunderstood nutrient. We're talking about vitamin K2.
And, the misunderstanding stems directly from how the vitamin was actually discovered.
I was reading about that. The history is fascinating.
Right. So in the late 1920s and early 1930s, researchers, guys like Henrik Dam and Edward Doisy, they identified this fat-soluble compound that was absolutely essential for blood clotting.
Okay, so that's where the clotting association starts.
Exactly. And they named it the coagulation vitamin, but they used the German spelling, so it started with a K.
Oh, wow. So that's literally why it's vitamin K, just the German word for coagulation.
Yep. That nomenclature effectively pigeonholed the nutrient for nearly a century. I mean, the global medical consensus basically viewed vitamin K as this single-purpose entity. Like, it was just the thing that stops you from bleeding to death if you suffer a major trauma.
Precisely.
Mm.
And it really wasn't until the late 20th century, and honestly the early 21st century, that advanced pharmacological data started coming out.
And these massive epidemiological trials, too.
Right. They revealed a completely bifurcated reality about this nutrient. It wasn't just one thing doing one job.
I've been looking through the stack of clinical reviews for today's deep dive, and the sheer volume of pleiotropic effects, meaning the diverse multi-system impacts this vitamin has, it's just staggering.
It really changes how you look at human nutrition.
We are moving so far beyond a basic clotting factor here.
Yeah.
I mean, we're talking about a molecule that fundamentally alters the electrical charge of specific proteins in your body.
It literally gives them the mechanical ability to bind, transport, and lock down minerals.
So today we're going to separate the localized clotting agent K1 from the systemic navigator, which is K2.
We have to unpack the atomic level chemistry of how it activates these proteins too. Yeah, and we'll dissect the massive Rotterdam and Neppen clinical trials. Plus, we are going to completely dismantle the pervasive medical myth that taking this quote-unquote coagulation vitamin will somehow turn your blood to sludge.
Well, to dismantle that myth, we first have to map out the biochemical family tree because, the vitamin K family is not just a monolith.
Right. It's a whole group of fat-soluble compounds.
Exactly. And they all share this specific core chemical structure, a 2-methyl-1-uro-4-naphthoquinone ring.
Okay, 2-methyl-1,4-naphthoquinone. Let's make that visual for the listener.
Sure. You can visualize that ring as the core engine of the vitamin. It's the active site where the electron transfer actually happens.
So the engine is identical across all forms of vitamin K.
Right. But while the engine is the same, the chassis, the side chain attached to that ring, is completely different depending on the form.
And that chassis dictates everything. Right?
It dictates its physical properties, how it's transported in the blood, and ultimately which tissues it is allowed to enter.
Okay, so the structural divergence starts with vitamin K1.
Also known as phylloquinone.
Phylloquinone, right, and K1 has what biochemists call a phytyl side chain.
Yes. And if we look at human dietary patterns, K1 is heavily represented.
Because it's synthesized by plants.
Exactly. Specifically in the chloroplasts, which means it is tightly bound to the photosynthetic process.
So when you eat dark leafy greens like spinach, kale, broccoli, collard greens, you are ingesting a ton of K1.
And the nutritional data on that is actually quite wild.
It really is. I mean, a standard half cup of cooked collard greens delivers about 530 micrograms of K1.
Which completely dwarfs the recommended daily intake.
Yeah. But this abundance of K1 in the diet is biologically logical when you consider where it actually goes.
Because of that specific fetal side chain, K1 is highly targeted. Once it crosses the intestinal barrier, it is almost exclusively taken up by the liver.
So it's basically a local specialist.
Right. The liver is the body's manufacturing hub for coagulation factors, specifically factors 2, 7IX, and X.
Okay, so K1 arrives in the liver, activates those specific clotting proteins, and its job is essentially done.
Yes. It has a very rapid turnover rate. It clears out of the bloodstream in just a matter of hours.
Which brings us to the real star of today's deep dive, vitamin K2.
Technically known as the menaquinones.
Menaquinones. And the
Yeah.
chassis on K2 is completely different from K1.
Totally different. Instead of a phytyl side chain, it has a polyisoprenoid side chain.
And K2 isn't just one molecule, right? It's a whole subfamily.
Yeah, it's classified by the length of that isoprenoid tail, ranging from MK4 all the way up to MK15.
And you definitely aren't getting these menaquinones from a spinach salad.
Definitely not. With the exception of MK4, which animals can synthesize in very small amounts from the K1 they eat,
Right.
the longer chain menaquinones are strictly the product of bacterial fermentation.
And that bacterial origin completely reframes how we have to look at cultural dietary habits.
It's a huge divergence. In Western diets, fermented foods are basically a supplementary novelty.
Yeah, like maybe some sauerkraut on a hot dog or certain aged cheeses like Gouda or Edam.
Which do contain modest amounts of K2. But the most robust source on the planet is a traditional Japanese dish called natto,
Right, natto, made by fermenting soybeans.
with a specific bacterium called Bacillus subtilis natto. The fermentation process yields just an astonishing concentration of the MK7 form of vitamin K2.
I saw the numbers on this. A small 3-ounce serving of natto contains roughly 850 micrograms of MK7.
It's incredible.
To put that 850 micrograms into perspective against a standard Western diet, let's look at a conventional chicken breast.
Okay.
That same 3-ounce serving of chicken yields about 13 micrograms of MK4.
13 versus 850.
Right. You would need to consume absurd, just astronomically impossible amounts of conventional Western meat to even approach the K2 levels found in a single spoonful of natto.
And this discrepancy isn't just a fun piece of trivia.
No, it is the epidemiological foundation for understanding why certain populations have vastly different rates of age-related arterial calcification.
Absolutely. Now, we should also briefly contextualize vitamin K3, or menaquinone.
Yeah, because it pops up in the literature sometimes.
It does, but we need to be clear. It is a synthetic water-soluble analog that is completely devoid of any side chain.
Meaning it doesn't have that chassis at all.
Exactly. And because it lacks that side chain, it actually interferes with cellular redox states. It generates reactive oxygen species, making it toxic to human liver cells at high doses.
So it's not something you want to supplement with.
Definitely not. It's relegated to animal husbandry and very specific laboratory research models, like exploring amyloid diseases.
Okay, so for human physiological optimization, the focus is entirely on naturally occurring K1 and the K2 menaquinones.
Correct.
I want to dig deeper into the physical mechanics of why that isoprenoid side chain on K2 changes its destination.
Let's do it.
If K1 and K2 have the same naphthoquinone engine, why is K1 trapped in the liver doing clotting work, while K2 gets a VIP pass to roam the entire circulatory system?
How does it reach the bones and the arterial walls?
Exactly.
It all comes down to lipophilicity.
Which is basically the molecular affinity for dissolving in fats, right?
Yes, and how the body packages lipids for transport. Because both forms are fat-soluble, their initial digestion requires a complex emulsion process.
So what happens when you actually swallow it?
When you consume them, your gallbladder releases bile salts, and your pancreas secretes lipases into the small intestine.
Right.
These enzymes break the dietary fats down into tiny micelles, which allows the vitamins to passively diffuse across the intestinal enterocytes.
So they pass through the gut wall.
Right. But this is a critical clinical note for you listening. Individuals with compromised pancreatic function or biliary insufficiency, or maybe people who have had their gallbladders removed,
They're going to have a hard time absorbing this
they face severe malabsorption hurdles for fat-soluble vitamins like K2.
because they can't break down the fats efficiently.
Exactly.
Okay, so once they cross that intestinal barrier, the sorting process begins,
Right. The enterocytes package the vitamins into chylomicrons.
which are basically just large lipid transport bubbles.
That's a great way to picture them. These chylomicrons travel through the lymphatic system and eventually empty into the venous blood.
And this is where the side chain length dictates the outcome.
Yes. As the chylomicrons circulate, enzymes strip away their triglycerides, leaving behind what we call a chylomicron remnant.
And the liver readily absorbs these remnants.
It does. Now, because K1 has a short, less lipophilic side chain, it stays in the liver.
It basically gets off at the first stop.
Precisely. However, the longer chain menaquinones, particularly MK7, are highly lipophilic. They don't just stay in the liver.
So what does the liver do with them?
The liver actively repacks MK7 onto low-density lipoproteins LDL and very low-density lipoproteins VLDL.
Which are the primary transport vehicles that travel outward from the liver to supply the peripheral tissues.
Exactly. The cholesterol transport system essentially becomes the superhighway for MK7 to reach the extrahepatic tissues, the bones, the cartilage, the vascular smooth muscle.
That is fascinating. The liver just uses the LDL cholesterol to ship the K2 out to where it's needed.
It's a beautifully elegant system, and the length of that side chain profoundly impacts the vitamin's half-life in the serum.
Which is the entire crux of why we focus on MK7 over MK4 for systemic supplementation, right?
Right. MK4, which has a short tail of just 4 isoprenyl units, has a biological half-life of roughly 1 to 2 hours.
That's incredibly fast.
It clears from the plasma so rapidly, behaving almost identically to K1 in that regard.
There's a landmark Japanese pharmacokinetic study detailed in our sources that perfectly illustrates the clinical reality of this half-life disparity.
No, it's a 7-day trial.
Yes. The researchers structured this trial with healthy female subjects. They administered identical physiological doses, 60 micrograms of MK4 to one group and 60 micrograms of MK7 to the other.
And they did this consistently for 7 days.
7 days of consistent low-dose supplementation.
The serum analyses from that study are just definitive.
What happened with the MK4 group?
In the MK4 group, the serum concentrations remained utterly flat. Even after 7 days, the MK4 levels in their blood were practically undetectable.
Wow.
They were registering below 16 picograms per milliliter. The molecule just cleared the system so rapidly that a daily physiological dose failed to establish any sort of baseline accumulation.
It was just gone.
Basically, yes.
Yeah.
Conversely, the group receiving 60 micrograms of MK7 exhibited a highly significant progressive increase in serum levels.
Because MK7 has a plasma half-life of 48 to 72 hours.
Exactly. It survives in the bloodstream for 2 to 3 days.
So day after day, that 60 micrograms compounded. It created this constant, stable reservoir of the vitamin bathing the peripheral tissues.
And this pharmacokinetic reality dictates clinical application. If a practitioner wants to elicit a systemic bone-building effect using MK4, they cannot rely on nutritional doses.
They have to go massive.
They are forced into pharmacological dosing. In Japan, MK4 is prescribed as an osteoporosis treatment at a massive dose of 45 milligrams a day.
Wait, 45 milligrams. That is 45,000 micrograms.
Right. Divided into 3 daily doses just to compensate for the rapid clearance rate.
But with MK7, because of its prolonged half-life, a mere 45 to 90 micrograms per day is sufficient to fully carboxylate the target proteins outside the liver.
Which brings us to the core molecular mechanism, carboxylation.
Yes, let's get into the clause. We've established that MK7 gets onto the LDL transport highway, circulates for days, and reaches the tissues.
But what is the menaquinone engine actually doing once it arrives at a bone cell or an artery wall?
Right. Vitamin K operates entirely as an obligatory cofactor for a specific enzyme called gamma-glutamyl carboxylase.
Frequently abbreviated as GGCX.
Let's break down the chemistry of GGCX without losing the narrative here.
Sure. So the body produces a family of proteins known as vitamin K-dependent proteins, or VKDPs.
Okay.
When these proteins are newly synthesized by the ribosomes in your cells, they are structurally incomplete.
They contain specific amino acid residues of glutamic acid, or GLU.
Right, exactly. And in this uncarboxylated state, the protein is completely inert. It literally cannot perform its biological function.
So the GGCX enzyme is responsible for activating them.
Right. But it cannot function without the reduced form of vitamin K, known as vitamin K hydroquinone.
So the vitamin comes in and unlocks the enzyme.
Yes. When the hydroquinone binds to the enzyme, it facilitates this remarkable structural modification. The enzyme extracts a proton from the gamma carbon of that glutamic acid residue.
Which allows a molecule of carbon dioxide to attach.
Exactly. This converts the glutamic acid, the GLU, into gamma-carboxyglutamic acid, or GLA.
The elegance of this transformation is just amazing. It lies entirely in the electrical charge.
It does.
So the original glutamic acid residue has one carboxyl group attached to it. The newly formed gamma-carboxyglutamic acid has two carboxyl groups right next to each other.
And in biochemistry, adding a second carboxyl group isn't just a structural footnote.
No, it completely alters the electrochemical properties of the protein.
It creates a highly electronegative pocket.
I visualize it as the protein suddenly sprouting a microscopic magnetic claw.
That is a perfect analogy.
Without K2, the protein is essentially smooth. It has no grip. But when K2 facilitates that carboxylation, adding the second carboxyl group, the protein suddenly possesses these strong, negatively charged molecular claws.
And because opposite charges attract, these negative claws are perfectly calibrated to grab onto divalent calcium ions, which carry a double positive charge.
Calcium ions are K+, so it's just basic magnetism.
This is the singular mechanism that governs calcium mobility in the human body. K2 is the switch that activates the claws, enabling the proteins to manipulate calcium.
However, this process oxidizes the vitamin K, right?
It does. The hydroquinone donates its electrons to power the carboxylation, and in doing so, it transforms into an inactive compound called vitamin K epoxide.
And if the process stopped there, we would exhaust our vitamin K supply in minutes.
The body would constantly be in a state of catastrophic deficiency.
Which is why evolution developed the vitamin K cycle.
Right. The body utilizes another enzyme, vitamin K epoxyreductase, or VKORC1.
VKORC1, what does that do?
This enzyme takes the inactive epoxide, reduces it, and basically recycles it back into the active quinone form, ready to bind with GGCX all over again.
So it is a highly conserved, closed-loop recycling system that maximizes the utility of every single molecule of vitamin K you ingest.
It's incredibly efficient.
Knowing this, we can now map this microscopic magnetic claw directly onto the grand pathology we opened with, the calcium paradox.
Yes, the dual disease state.
If a person is chronically deficient in vitamin K2, which, based on the shift away from fermented foods and grass-fed dairy, a massive portion of the Western world is face, their extrahepatic proteins never get their claws.
We must apply this directly to the vascular system, specifically focusing on a protein called Matrix Gla Protein, or MGP.
Okay, MGP.
MGP is synthesized directly by the vascular smooth muscle cells that make up the structural walls of your arteries.
So it lives in the artery wall.
Yes, and its biological mandate is to actively inhibit the precipitation and crystallization of calcium phosphate within the arterial tissue.
It is the ultimate defense mechanism against calcification.
Absolutely. But MGP is a vitamin K-dependent protein.
Meaning it needs those claws.
If there isn't enough circulating MK7 to activate the GGCX enzyme in the vascular tissue, the MGP remains undercarboxylated.
It's called UCMGP.
Right. It lacks the double carboxyl groups, it has no claws, and it is entirely powerless to bind the calcium ions that are just drifting through the bloodstream.
And the downstream pathology of inactive MGP is just horrifying at a cellular level.
It really is. Without functional MGP clearing the microcrystals of calcium, the vascular smooth muscle cells undergo what we call a phenotypic switch.
They experience transdifferentiation.
They literally lose their vascular identity and transform into osteoblasts-like cells.
So the artery literally becomes confused and begins actively laying down bone matrix inside the blood vessel.
The artery is ossifying. The tunica media, the middle muscular layer of the artery, turns into a rigid pipe.
And this leads to severe systolic hypertension, right?
Yeah.
Because the heart is forced to pump against the stiffened vascular tree.
Exactly, which ultimately results in left ventricular hypertrophy. The heart muscle enlarges dangerously, just trying to push blood through these bony tubes.
The epidemiological evidence confirming MGP's reliance on K2 is definitive. We see it most notably in the Rotterdam study.
The Rotterdam study is a cornerstone piece of data.
This was a massive, prospective, population-based cohort study. Researchers tracked 4,807 subjects, men and women over the age of 55, who had no history of myocardial infarction at the baseline.
And they tracked them meticulously for 7 to 10 years.
Using detailed dietary questionnaires and advanced radiological imaging to actually quantify aortic calcification.
Right.
The researchers divided the cohort into tertiles based on their daily intake of vitamin K2.
Meaning they split them into 3 groups: low, medium, and high intake.
Exactly. And the statistical divergence between the highest intake group and the lowest intake group is staggering. Subjects in the upper tertile of K2 intake exhibited a 52% lower risk of severe aortic calcification.
52% is a massive risk reduction.
And that translates directly to mortality outcomes. That same high intake group had a 57% lower risk of dying from cardiovascular disease.
And a 26% lower risk of all-cause mortality just from having high K2.
What makes the Rotterdam data so pivotal is the control variable. The researchers also tracked vitamin K1 intake across the same exact cohort.
And what did they find?
High dietary intake of K1 showed absolutely no correlation with reduced cardiovascular risk or reduced calcification.
Not at all.
The cardiovascular protection was exclusively tied to the menaquinones.
Which perfectly proves the pharmacokinetic theory we discussed earlier. K1 remains trapped in the liver, while K2 provides the systemic vascular defense.
Now, the Rotterdam study is observational, which always invites questions about confounding variables. People listening might wonder, maybe the people eating more K2 were just generally healthier in other ways.
It's a fair question, but we have interventional data that proves causality.
The Napin trial is a perfect example of this.
Napin and colleagues designed a double-blind, randomized, placebo-controlled trial, the absolute gold standard of clinical research.
They took 244 healthy postmenopausal women and administered a specific nutritional dose of 180 micrograms of MK7 daily for 3 full years.
And they measured arterial stiffness using pulse wave velocity.
Which traps how fast the pressure wave from a heartbeat travels through the arteries.
Right. Faster waves indicate stiffer arteries.
So what were the results over the 3 years?
They were incredible. In the placebo group, pulse wave velocity increased, demonstrating the progressive arterial stiffening that the medical community generally accepts as a quote-unquote normal part of aging.
But in the group receiving the 180 micrograms of MK7.
Not only did the calcification halt, but the pulse wave velocity actually decreased.
Oh, wow.
The subjects exhibited a measurable improvement in arterial elasticity.
They literally reversed the age-related stiffening of their vascular tissue simply by providing the cofactor needed to activate MGP.
This introduces a profound clinical application. Data discussed in forums like MedPage Today indicate that while MK7 supplementation cannot magically dissolve heavily calcified, advanced atherosclerotic plaques.
Because once the tissue has fully ossified, the damage is structural.
Exactly. But it is remarkably effective at arresting the progression of early to moderately developed non-calcified plaques.
By fully carboxylating the MGP, it creates a chemical blockade, preventing further calcium deposition.
Right.
But since K2 is actively blocking calcium from depositing in the tunica media of the artery, that calcium remains suspended in the serum.
The body has to put it somewhere to avoid hypercalcemia.
Which brings us to the osteoblasts and the other half of the calcium paradox. We know the vascular defense protein is MGP. The skeleton's counterpart is osteocalcin.
Osteocalcin is the most abundant non-collagenous protein found in the bone matrix.
And it is secreted by osteoblasts, which are the specialized cells responsible for synthesizing new bone.
Just like MGP, osteocalcin is fundamentally dependent on vitamin K2 for carboxylation.
If the osteoblast secretes uncarboxylated osteocalcin, the protein just drifts into the extracellular space.
Entirely incapable of binding the calcium floating by.
The calcium cannot integrate into the hydroxyapatite matrix, which is the hard mineralized lattice of the bone.
But when MK7 arrives and the GGCX enzyme carboxylates the osteocalcin,
those magnetic claws engage.
Exactly. The carboxylated osteocalcin violently binds the circulating calcium ions and physically anchors them into the hydroxyapatite lattice.
It is literally the molecular cement that dictates bone mineral density.
And the literature supporting this mechanism is vast. A comprehensive meta-analysis published in the journal Frontiers aggregated data from 16 randomized controlled trials,
encompassing over 6,400 postmenopausal women.
The synthesis of that data confirmed that vitamin K2 supplementation significantly increases lumbar spine bone mineral density
and dramatically reduces the incidence of osteoporotic fractures.
This brings us back to the Japanese osteoporosis protocol, which I find so fascinating because of the sheer scale of the dosing.
We mentioned earlier that MK4 clears the blood rapidly, requiring massive doses to achieve a systemic effect.
The Japanese Ministry of Health approved MK4 as a pharmaceutical intervention at 45 milligrams per day.
When they studied this aggressive pharmacological dosing in older patients with diagnosed osteoporosis over a 2-year period, the clinical outcomes were almost hard to believe.
The 45 milligram protocol reduced the incidence of vertebral fractures by an astounding 50%.
Dropping the fracture rate from 30% in the control group down to 14% in the MK4 group.
It proves that supplying the requisite cofactor to activate osteocalcin fundamentally changes the structural integrity of the skeleton.
I was reading about how this interacts with standard Western osteoporosis treatments, specifically bisphosphonates like alendronate.
Right.
Bisphosphonates work by inducing apoptosis cell death in osteoclasts, which are the cells that break down bone.
So the drug stops bone loss, but it doesn't actively build new, highly mineralized bone.
That is the crucial distinction. Bisphosphonates inhibit resorption. Vitamin K2 facilitates mineralization.
And when researchers combine the two therapies, the results are highly synergistic.
The patient benefits from the anti-resorptive properties of the bisphosphonate, while simultaneously utilizing the K2 to aggressively carboxylate osteocalcin.
Driving calcium into the newly preserved bone architecture.
It perfectly illustrates the flaw in the simplistic take a calcium pill mentality. I really love the construction site analogy here.
It's a great way to think about it.
If you are building a brick wall, the calcium represents the bricks. Dumping a massive load of bricks at the site doesn't build the wall, it just creates a dangerous pile.
Exactly.
The bisphosphonate might stop a bulldozer from knocking down the existing wall, but it doesn't lay new bricks. Vitamin K2 is the master mason. It picks up the bricks and meticulously cements them into the foundation.
Extending that analogy, if K2 is the mason, vitamin D3 is the foreman who orders the bricks and orchestrates their delivery to the site.
Oh, absolutely. The biochemical synergy between D3 and K2 is arguably the most critical nutrient pairing in human endocrinology.
This is where we need to look at your supplement cabinet. Millions of people routinely supplement with high doses of vitamin D3, especially during the winter months.
Aiming to support immune function or mood, mostly.
Right, but they are completely unaware of the downstream calcium cascade they are triggering.
To understand the danger, we must look at D3's primary mechanism of action regarding minerals.
Vitamin D3, once converted to its active hormonal form, calcitriol, enters the nucleus of the enterocytes in your intestines.
And it aggressively upregulates the synthesis of calcium-binding proteins, specifically calbindin.
It essentially opens the cellular floodgates. It maximizes the absorption of dietary calcium across the intestinal wall and into the systemic circulation.
So vitamin D3 guarantees you have an abundance of calcium in your blood.
Yes, but vitamin D3 possesses absolutely no mechanism to direct that calcium.
It doesn't tell the calcium to go to the skeleton instead of the aorta.
It creates a massive biological liability if K2 is absent.
If you artificially spike your serum calcium levels with high-dose D3, but you possess a deficiency in K2, your MGP remains uncarboxylated.
Your osteocalcin remains uncarboxylated.
The calcium just wanders aimlessly, and thermodynamic principles dictate it will precipitate in the soft tissues.
The clinical data on this specific dual deficiency is frightening.
Studies tracking patients deficient in both vitamin D and vitamin K2 reveal a 64% increased risk of all-cause mortality compared to those with optimal levels of both.
The hypercalcemia drives severe adverse cardiac remodeling. You are essentially fueling the transdifferentiation of your arteries.
It is a self-inflicted calcium paradox.
Furthermore, to complete the physiological picture, we must acknowledge magnesium.
Magnesium serves as the requisite cofactor for the binding of ATP adenosine triphosphate.
The initial enzymatic conversion of vitamin D in the liver, its subsequent activation in the kidneys, and the mechanical function of the GGCX enzyme all require ATP.
Without adequate magnesium, the energetic currency required to run the D3 and K2 pathways simply isn't available.
The triad of D3, K2, and magnesium represents the foundational axis of calcium homeostasis.
So we've established K2 as the master traffic cop for calcium, protecting the bones and the heart. But the scientific literature over the last 5 years has begun tracking K2 into entirely new biological territory.
Far beyond calcium regulation.
We are looking at pleiotropic effects in the central nervous system.
The neurological frontier is profoundly exciting. In the brain, vitamin K2 intervenes directly in the synthesis of a highly complex class of lipids known as sphingolipids.
I know sphingolipids are heavily concentrated in the cell membranes of the central nervous system.
They are the primary structural components of the myelin sheath.
The fatty insulation that wraps around the axons of your neurons, allowing electrical impulses to travel efficiently without short-circuiting.
Right. But how does a vitamin meant for bone calcium suddenly start regulating brain fat?
It speaks to the evolutionary efficiency of these molecules. The MK4 form of vitamin K2 is found in remarkably high concentrations in the mammalian brain.
The current biochemical models suggest that K2 activates specific enzymes within the endoplasmic reticulum of brain cells.
Enzymes that dictate the rapid turnover and metabolism of these sphingolipids.
By maintaining the structural integrity of the myelin sheath, K2 actively supports neuronal proliferation, differentiation, and overall survival against age-related cognitive decline.
And beyond the structural insulation, there is an even more aggressive neuroprotective mechanism outlined in our sources. anti-ferroptotic activity.
Ferroptosis is a mechanism I hadn't encountered before diving into this research either.
It's a specialized, highly destructive form of programmed cell death.
Unlike apoptosis, which is a clean, organized dismantling of a cell, ferroptosis is driven by iron-dependent lipid peroxidation.
Let's break down lipid peroxidation.
The cell membranes of your neurons are rich in polyunsaturated fatty acids.
In the presence of excess intracellular iron, and under conditions of oxidative stress, a vicious chemical chain reaction called the Fenton reaction occurs.
It generates massive amounts of reactive oxygen species. These free radicals attack the fatty acids in the cell membrane.
Literally oxidizing and destroying the cellular envelope.
And a primary driver of this peroxidation is an enzyme called 12-lipoxygenase, or 12-LOX.
And this is where the specific structure of vitamin K enters the picture, not as a protein carboxylator, but as an antioxidant.
Precisely. Researchers have discovered that the fully reduced form of vitamin K2, the hydroquinone, acts as a uniquely potent, radical-trapping antioxidant in the neural environment.
It specifically targets and inhibits the 12-LOX enzyme.
By neutralizing 12-LOX and scavenging the reactive oxygen species, the K2-hydroquinone completely halts the lipid peroxidation chain reaction.
It stops the neuron from dying via ferroptosis.
The implications for neurodegenerative diseases are massive.
If ferroptosis is a primary driver in the neuronal death associated with Alzheimer's or Parkinson's, and K2 actively blocks that pathway, we're looking at a fundamental neuroprotective agent.
The sources also point to K2 assisting in amyloid clearance and even modulating insulin sensitivity to protect metabolic health.
It is the absolute cutting edge of anti-aging research.
We must temper that excitement with clinical reality, though.
Right.
The mechanisms of ferroptosis inhibition and sphingolipid synthesis are proven in vitro and in animal models.
The bone density and cardiovascular data we discussed earlier are built on decades of massive randomized human trials.
The neurological data is the emerging frontier. It requires vast longitudinal human trials to establish specific therapeutic dosing protocols for dementia or metabolic syndrome.
But the mechanistic foundation is undeniable.
It really is.
With all of these incredibly powerful systemic effects, pulling calcium into bone, stopping arteries from calcifying, halting neuronal cell death, we arrive at the most pressing question for anyone considering supplementation.
What is the toxicity limit?
Exactly. And does the name coagulation vitamin mean that high doses will cause dangerous pathological blood clots?
It is the single most persistent myth in nutritional science, and we must dismantle it using hepatic physiology.
In a healthy individual, supplementing with high doses of vitamin K2 does absolutely nothing to cause excessive blood clotting or thick blood.
It will not induce a thrombosis.
But it explicitly activates the coagulation factors in the liver. How does more K2 not equal more clotting?
It is dictated by a strict biological ceiling known as a saturation point.
Okay, saturation point.
The liver synthesizes a finite, genetically predetermined number of inactive coagulation proteins, factors 2, 7, 9, and X.
When you ingest vitamin K, the liver uses it to carboxylate those specific proteins.
However, once 100% of those available proteins are carboxylated, the system is fully saturated.
I think the gas tank analogy is the most intuitive way to grasp this. Think of your liver's clotting capacity as a 15-gallon gas tank.
Okay.
Because preventing a fatal hemorrhage is an evolutionary priority. The body will always route the first available vitamin K to fill that specific tank.
Right.
But once you hit 15 gallons, the tank is completely full. The pump automatically shuts off. You cannot squeeze 20 gallons of coagulation into a 15-gallon tank.
Exactly. The enzymes simply have no more substrate to work on.
The coagulation factors operate at normal, healthy homeostasis.
Any excess vitamin K2 you consume bypasses the fully saturated liver. It enters the LDL transport highway we discussed earlier and is delivered to the extrahepatic tissues to power the MGP in the arteries and the osteocalcin in the bones.
Because of this strict saturation point, the toxicity profile of vitamin K is almost non-existent in healthy populations.
Major international regulatory bodies, including the European Food Safety Authority and the U.S. National Institutes of Health, have completely failed to establish a tolerable upper intake level AUL for vitamin K.
There simply isn't an observable toxic threshold from dietary or supplemental intake.
The Council for Responsible Nutrition has published data confirming exceptional safety even at continuous doses exceeding 1,000 micrograms per day.
However, there is one absolute life or death contraindication that every listener must understand.
While K2 is entirely safe for healthy individuals, it presents a severe, potentially fatal interaction for patients actively taking vitamin K antagonists.
Specifically, the oral anticoagulant warfarin, commonly known by the brand name Coumadin.
We have to analyze the pharmacology of warfarin to understand why this interaction is so explosive.
Warfarin is prescribed to patients who have mechanical heart valves, atrial fibrillation, or a history of deep vein thrombosis, people who are at an acute risk of a stroke if their blood clots normally.
We discussed the vitamin K recycling cycle earlier, specifically the VKORC1 enzyme that reduces the inactive epoxide back into the active hydroquinone.
Warfarin acts as a direct competitive inhibitor of that specific VKORC1 enzyme. It physically blocks the recycling process.
By halting the recycling, warfarin forces the liver into a state of severe artificial vitamin K deficiency.
The liver quickly runs out of active hydroquinone, meaning it can no longer carboxylate the coagulation factors.
The blood becomes artificially thin, preventing the formation of pathological clots.
Cardiologists monitor this precarious state using a metric called the INR, the International Normalized Ratio.
A normal person's INR is 1.0. A patient on warfarin is typically maintained between 2.0 and 3.0, meaning their blood takes 2 to 3 times longer to clot.
If a patient stabilized on warfarin goes to the pharmacy and purchases a high-dose vitamin K2 supplement, they initiate a catastrophic biochemical override.
The supplement floods the hepatic portal vein with massive amounts of fresh, unoxidized ketones.
Because these ketones are fresh, they completely bypass the need for the blocked VKORC1 recycling enzyme.
The liver suddenly has an unrestricted supply of active cofactor.
It instantly resumes the massive production of carboxylated clotting factors.
The patient's INR crashes from a therapeutic 2.5 down to 1.0 in a matter of hours.
The blood thickens back to normal, entirely obliterating the protective effect of the warfarin.
That patient is now in immediate critical danger of suffering a massive stroke or pulmonary embolism because their underlying cardiovascular condition is no longer being chemically managed.
The sensitivity is profound. Literature cites case studies where patients experienced dangerous drops in their INR simply by switching to a daily multivitamin that contained a seemingly negligible 25 micrograms of vitamin K.
Furthermore, the inverse scenario is equally lethal.
If a patient secretly takes K2, causing their doctor to prescribe a massive dose of warfarin to fight it, and the patient suddenly stops taking the supplement,
The massive warfarin dose goes entirely unopposed.
the INR skyrockets, and the patient risks a fatal internal hemorrhage.
The absolute rule is, if you are on a vitamin K antagonist, you do not touch a vitamin K supplement without direct, continuous supervision from your cardiologist.
However, diving into the advanced hematology research, there's a fascinating paradox regarding warfarin and low-dose K2.
It is a counterintuitive mechanism, but heavily documented.
Patients on warfarin often suffer from wildly erratic INRs. Their blood is too thick one week and too thin the next.
This instability is frequently driven by dietary fluctuations. If a patient eats a large spinach salad on Tuesday, they flood their system with K1, dropping their INR.
If they avoid greens for the rest of the week, the K1 clears and their INR spikes.
To combat this dietary whiplash, some clinical protocols, including research highlighted by Life Extension, involve administering a very low, strictly controlled daily dose of vitamin K.
Typically between 50 and 150 micrograms.
The logic is to establish a permanent, immovable baseline of vitamin K in the serum.
By providing a constant low dose, the doctor can carefully titrate the warfarin dosage against that specific baseline.
It dampens the impact of day-to-day dietary changes, resulting in a significantly more stable and predictable INR.
But again, this protocol requires meticulous, continuous phlebotomy and physician oversight. It is not a biohacking experiment to try at home.
We also need to clarify the status of newer anticoagulant medications, the DOAC's direct oral anticoagulants, such as apixaban, Eliquis, or rivaroxaban, Xarelto.
The pharmacology of DOACs is fundamentally different. They do not target the VKRC1 enzyme.
They do not interfere with the vitamin K cycle at all.
Instead, they directly bind to and inhibit highly specific, already activated components of the coagulation cascade, such as factors Ager or thrombin.
Because their mechanism of action is entirely divorced from carboxylation, DOACs do not interact with vitamin K.
Patients taking modern DOACs can safely utilize vitamin K2 supplementation without compromising their anticoagulant therapy.
This deep dive fundamentally alters the landscape of how we view mineral metabolism and longevity.
Let's trace the molecular journey one last time. We separated the localized, rapidly clearing clotting agent K1 from the systemic lipoic navigator K2.
We saw how the elongated isoprenoid tail of MK7 allows it to ride the LDL cholesterol highway for days.
Bathing the peripheral tissues in the necessary cofactor.
We examined the atomic elegance of the GGCX enzyme, watching it attach that second carboxyl group to convert inert proteins into active calcium-binding machines with powerful electronegative claws.
Those claws are the definitive solution to the calcium paradox.
They allow Matrix Gla Protein to fiercely defend the tunica media of your arteries, stopping vascular transdifferentiation and actually reverting arterial stiffness, as proven by the Nappen pulse wave velocity data.
Simultaneously, those same claws allow osteocalcin to capture the calcium from the serum and physically cement it into the hydroxyapatite matrix of the skeleton,
Significantly reducing osteoporotic fractures and operating synergistically with standard anti-resorptive therapies.
We established the absolute necessity of pairing high-dose vitamin D3 with K2 and magnesium, ensuring that the flood of intestinal calcium induced by D3 is safely directed away from the heart and into the bones.
We explored the cutting-edge neuroprotective properties of K2-hydroquinone, neutralizing 12-LOX to prevent lipid peroxidation and ferroptosis in the brain.
And we completely debunked the myth of K2 causing thick blood, proving that the liver's strict saturation point makes K2 incredibly safe for everyone, except those actively managing VKRC1 blockades with warfarin.
It is a profound paradigm shift. But to truly synthesize this information, we have to look beyond our biochemistry and look at the macro environment that caused this widespread deficiency in the first place.
I want to leave you with a final thought regarding the invisible biochemical chains that link our health to agricultural practices. We established that animals synthesize MK4 by converting the K1 they ingest.
Right, cows eat grass, which is loaded with K1 from the chloroplasts, and their biology converts it into the K2 found in their milk and meat.
Historically, yes. But over the last century, industrial agriculture fundamentally altered that equation.
We removed our livestock from green K1-rich pastures and isolated them in concentrated feedlots.
We replaced their evolutionary diet of green grass with highly subsidized, efficiently grown grains like corn and soy.
Corn and soy possess virtually no vitamin K1.
The raw material is gone.
The animal cannot convert what it does not consume. By altering the diet of our livestock to maximize caloric efficiency and profit, we unknowingly severed a critical link in the nutrient chain.
We effectively engineered this vital, life-saving, calcium-directing molecule out of the modern Western food supply.
Next time you evaluate your diet, or consider why a supplement might be biologically necessary, think about that unseen chain of nutrients.
The calcification of our arteries and the weakening of our bones isn't just a consequence of aging. It is a direct reflection of a biochemistry that started in the soil and was broken by industrialization.
That is an absolutely chilling and brilliant realization. We broke the chain at the source.
It brings us perfectly back to the beginning of our discussion. The standard medical framework is often too constrained, treating the rigid artery and the brittle bone as separate, isolated failures of aging.
But when you step back and apply the lens of nutritional biochemistry, you see the interconnected reality.
You understand the invisible traffic cop that has been missing from the intersection. Tree health isn't just about flooding the system with calcium and hoping for the best. It's about providing the specific cellular masons required to build the foundation.
Check your supplement labels, evaluate your D3 intake, and consider how the biochemistry of your food has changed.
Thank you for joining us on this deep dive. Keep asking the hard questions, look past the obvious symptoms, and always stay curious.